third day of the Barclays Global Healthcare Conference. My name is Jeff Meacham. I'm the Senior Biopharma Analyst. And we're thrilled to have Moderna this morning present. And speaking on behalf of Moderna, we have Stephen Hoge, who's the President, and Lorence Kim, CFO. Guys.
Thank you, Jeff. I'm Lorence Kim, CFO of the company. Let me start off the presentation, and then I'll hand it over to Stephen, our President, to take us through the bulk of the programs that we have. So, and I'll also start by saying that we will be making forward-looking statements in this presentation. You can find risk factors in our S1 filing and our other SEC filings. So, Moderna was founded on this belief that messenger RNA would form the basis for a new drug class. At its core, Moderna we believe that mRNA is the software of life, that it forms a basis for instructing cells to express protein. And what you see on this slide is a bit of an illustration of why the company began down the path that it did 8 years ago, which is we saw an extraordinary opportunity in front of us because we believed we could get mRNA into cells and we can get the native cellular machinery in the form of ribosomes to translate our proteins for us. We could direct those proteins to a whole variety of cellular compartments that were previously inaccessible to drug-like modalities, including membrane proteins, mitochondrial proteins, nuclear proteins even. And on the basis of that, what was really evident to us was that the opportunity was enormously large here. We had a tremendous white space potentially available to us. On top of that, because messenger RNA could work as a technology platform, meaning that the delivery systems could be utilized somewhat universally across the portfolio because the mRNA technologies and process technologies could be leveraged. We believe that by de-risking sequentially this portfolio, we would ultimately end up with a higher probability of technical success as we progress. Further to that, we believe that the research and development process could be significantly accelerated because of these shared features, because of the shared technologies. And then lastly, we saw an opportunity for significant capital efficiency, in particular because things like our manufacturing process ends up getting shared across the portfolio because it gets scaled up very easily because it's ultimately a cell-free process. And so we believe that would also create a significant advantage for us if we were to do the development of this drug class at scale. Now, what we've seen so far is evidence of these features. We have 20 product candidates in the portfolio right now. We have been de-risking the portfolio with a whole series of clinical data readouts thus far, including having 1,000 subjects in clinical trials to date. We've been on an accelerated path. Those 20 development candidates have been realized over the course of 4 years, and so that's an extraordinary pace. And then the capital efficiency has really been showing itself in terms of our efficiencies that we've been realizing and continuing to see, especially with the coming online of our Norwood manufacturing facility last year. Now, to realize this opportunity in front of us, we've really focused on risk management. It's something that you should know about us, which is as we've been developing this technology, we've done a number of things. We've been trying to disaggregate risk. And in particular, we've been separating out what we view as technology-based risk versus biology risk or disease risk. And the way that we segment our technology risk has been in the form of what we call modalities. These modalities are groups of product candidates that all share product features. And you can see the 6 that we currently have in development here. I won't go through them at this point, but I will say that they all have shared underlying technologies that allow for de-risking within each of these vertical areas. And what we've done in our portfolio development is that we've attempted to sequentially go through these modalities. And then also within the context of each modality, we've elected to manage the disease biology risk by going after more tangible and simpler candidates first. And the best example of that is some of our first programs that we did, which were flu vaccines. Why do we do that? Flu has the incredible benefit of being able to show us clinical proof of concept early. Flu vaccines are approved on the basis of very clear quantitative neutralizing titers, and we were able to demonstrate that a number of years ago in our first foray into the clinic. And from there, once we demonstrated success, we were able to then move on rapidly to more complicated disease biology, including advancing what we have now in the clinic in the form of a CMV vaccine, which is a more complicated set of antigens, but correspondingly significant commercial opportunity. And we also went from infectious disease and transitioned to cancer vaccines in collaboration with Merck, and we have in development, starting in phase 2 quite soon, a personalized cancer vaccine, which we'll talk about in just a bit. Correspondingly, in other verticals, we've progressed in similar ways. And so in the middle two modalities that you see here in the intratumoral setting and the localized regenerative therapeutic setting, these are all variations of directly injecting tissues with mRNA product candidates. And there we started with a program called VEGF, which is partnered with AstraZeneca. That's currently in a phase 2. But we got our proof of concept in a phase 1, demonstrating not just safety and tolerability, but also protein expression and pharmacology. And we've been advancing since then. We've been advancing in an intratumoral setting where in a similar fashion we are injecting into tumors and expressing relevant immunologically active proteins like the OX40 ligand program and what we term the triplet. And so you see this progression playing out in other parts of the portfolio. The last piece of that progression is on the far right, where you see these systemic intracellular and systemic secreted therapeutic modalities. And here we are in the midst of our first clinical experiences. The first program in these verticals is the chikungunya antibody program, where we've just initiated dosing and have cleared the first dose cohort in this setting, where we are using this program, one, to see if we can elicit passive immunity against a terrible virus in chikungunya, but also to demonstrate that we can translate significant amounts of protein in humans in the form of IgGs that are easily quantifiable. And from there, what you're going to see out of us is a whole series of additional programs, including representative programs on this slide, like methylmalonic acidemia, which has an open IND now, and the Fabry program. Again, demonstrating our progression as we try to disaggregate this risk. So where are we right now? I'll just spend a second on execution. I think that's the big story for us for the next couple of years. For 2019 and 2020, we did announce that our priorities are really first and foremost on executing on the current development pipeline, where we have these 20 programs in development. We are very focused on driving clinical proof of concept data, and you'll see some of that already coming to fruition. And then second and thirdly, we are going to be really continuing to expand our pipeline with new development candidates in our existing modalities, so the 6 modalities that you saw on the prior slide. And third, we continue to invest to bring forward new development candidates in new modalities. We are not done trying to innovate, and so we continue to invest in the opportunity to go into new tissue types, new routes of administration, and we will talk more about that when those are ready. So to give you a sense for what we've been up to in terms of execution, this is a little bit of a snapshot of our development pipeline. We've been quite excited over the past couple of announcements that we've made to demonstrate really significant progress across our entire portfolio. In particular, the 7 highlighted programs here are all significant advancements since the IPO about 3 months ago. And so I'll just touch on them briefly here. Stephen will go into further detail, but for the personalized cancer vaccine program on the far right, and OX40 ligand, we are advancing in phase 2 planning. For the HMPV PIV3 vaccine, sort of in the middle of this page, we announced positive phase 1 data just a few weeks ago. For the chikungunya antibody and the triplet program, we initiated phase 1 dosing and have moved through the first cohorts in each of those programs. And for the methylmalonic acidemia and IL-12 programs, we were able to get open INDs with the FDA. The IL-12 program is under the control of AstraZeneca. And so to us, this illustrates this point that we are built to execute across this very broad portfolio. The last piece of execution that I'll touch on here is our manufacturing site. And so this is a snapshot of our Norwood, Massachusetts facility. We've been working on this for a couple of years. It was opened last summer. And this is really an enabling investment for us. further to my point earlier about capital efficiency, we view this as an important piece of allowing us to strategically control our supply chain as a development-focused manufacturing facility where we could combine and vertically integrate a whole series of capabilities, including the manufacturing of mRNA, the formulation of that mRNA, the filling of vials, labeling, and ultimately now we're actually continuing to consolidate on critical raw materials and bring those in-house. We also have in this site our personalized cancer vaccine manufacturing unit, which we brought in where it was previously sitting outside of Moderna at a CMO, and we also have here what we've previously talked about, which is an automated preclinical production site. This used to be housed in Cambridge, but this is our facility that enables us to mass produce up to 1,000 mRNAs per month for the use of research. So this is a critically important site, and we invite folks to come by and visit. We will frequently host tours of this site. So with that, let me pass it over to Stephen to take you through some of the progress in our modalities.
Great. Thanks, Lawrence. So I have the enviable task of going through 20 programs in 10 minutes. So I'm going to go pretty quick and cover these in the same way that we think about them, which is modalities, and that was what Lawrence was just describing, the 6 across the page. So the most advanced in terms of number of programs is our prophylactic vaccines modality. As Lawrence described, we have 5 positive phase 1 results already, and Merck is preparing for their phase 2 in RSV. Over 950, about 950 subjects dosed in studies to date, and a pretty clear emerging safety and tolerability profile that is consistent with approved adjuvanted vaccines, any number of approved adjuvanted vaccines. We've shown tolerability up to doses of 300 micrograms. And if you look for activity, all the 5 phase 1 readouts that are described here ultimately met their primary endpoints in terms of activity. So the just recently reported HMPV/PIV3 combination vaccine, which we reported 4 weeks ago, was able to drive substantial neutralization titers of 3-fold and 6-fold in the case of HMPV. At all dose levels tested. The RSV vaccine previously reported by ourselves, Merck has already initiated planning for phase 2A development there. And then 3 of our earlier experiences, the chikungunya vaccine, where we saw 100% seroresponse in subjects at the top dose tested, and the 2 flu vaccines where we were able to achieve the approvable correlate of protection of 1 to 40 HAI titers in 96 and 100% for 2 different strains of those vaccines. Major next steps that we're anticipating across that portfolio, obviously the CMV cytomegalovirus vaccine is a large commercial opportunity that we're quite excited about. We're looking forward to the phase 1 safety and immunogenicity data, as well as the initiative planning for phase 2. HMPV/PIV3, as we announced when we announced the phase 1 positive results, the next step for that program will be an age de-escalation into the target, towards the target population, and so these are ultimately for children, and we're following a relatively standard process there going into phase 1B. RSV, I previously mentioned, Merck is planning for the phase 2A, and then the Zika vaccine, this is our second Zika vaccine, 1893, is partnered with BARDA, who is funding this work. We're looking forward to the IND filing of that vaccine in the future. So that's prophylactic vaccines. The next modality, as Lawrence described, that uses very similar technology, vaccines after all, is the cancer vaccines work. And in the case of cancer vaccines, what we're trying to do here, rather than express a viral antigen to develop an immune response to it, what we're trying to do is express a tumor neoantigen. In this case, we personalized it, and so we're actually making individual vaccines for individual patients who are suffering from cancer and putting in neoantigens that are specific for their cancer. The study is up and running, and we've actually enrolled and dosed over 30 patients, and we've actually dosed up to 1 milligram in that with no dose-limiting toxicities to date. I'll remind you that the design of the study includes arms that are both as monotherapy and in combination with Keytruda, as this work is being funded and co-developed with Merck. We have demonstrated activity. I'm showing one example of a patient in the 0.13 milligram cohort showing antigen-specific T-cell responses. And so what you're looking at is a range of different pools that are pulsed into this patient's blood that are showing specific primed and boosted responses. And as you look across the cycles, they're growing over time. in T-cell responses to the antigens in that patient's tumor. Again, we made a vaccine specifically for that patient. Anticipated next steps. Based on this and other data with Merck, we've actually started planning for a randomized phase 2 study, and I'll say more about that in just a second on the slide. We do have another program in the cancer vaccine space, also partnered with Merck. It was the subject of a $125 million deal about a year ago, and that is against KRAS specifically. This is not a personalized vaccine. This is a shared antigen vaccine for the most common mutations in KRAS. Merck is going to be running that clinical study, and the anticipated next step there would obviously be the start of that phase 1 study and hopefully soon thereafter T-cell immunogenicity data. So just a moment on the PCV program and its next steps. So we have actually filed the phase 2 protocol with the FDA, as we disclosed recently. This will be a randomized phase 2 personalized cancer vaccine plus Pembro versus Pembro alone in a high-risk melanoma patient population. What we're looking to do is assess directly whether postoperative adjuvant therapy with our vaccine plus Pembro improves recurrence-free survival. This is on the back of approvals in this indication that Pembro has already achieved. And given that, we were able to do this as a 2-to-1 randomization, where we'll have 2 patients randomized to the vaccine arm and 1 to the placebo control arm, and it's about 150 patients overall, about 101 arm and 50 in the other, as you can see there. And we'll be looking relatively quickly for recurrence-free survival, the primary analysis happening in about 12 months, and that's going to leverage a lot of the experience that recently Merck published about a year ago, and that was a subject to their approval of the FDA in that indication. So we're not going to spend many years trying to demonstrate activity. We're actually going to go take the direct randomized challenge study, or randomized head-to-head study as the next step, which is very exciting. That's the personalized cancer vaccine progress. Next modality is our intratumoral immuno-oncology modality. Here we're injecting directly into tumors, that's the intratumoral component, and expressing proteins that activate the immune system. And the goal is to drive a local immune activation against the cancer that might create an abscopal effect. This is precedented. There's approved drugs. T-VEC is an example of that, obviously from Amgen. We have 3 programs that are in development, 2 that are actually in clinical testing, and 1 that has an open IND with AstraZeneca, as you can see here. The OX40 program is now in a dose confirmation stage. We have dosed up to 8 milligrams, selected that as that dose. As had been disclosed in the S1, we actually have amended that protocol and filed to move forward with a phase 2 cohort in advanced ovarian carcinoma based on early signals in that phase 1 study. And so of the 2 patients that had advanced ovarian carcinoma in the phase 1 study, we saw local tumor regressions, lesion regressions in the injected lesions. They did not meet RECIST criteria, but they were sufficiently specific responses, and we also talked about the local expression of the protein in some of these, in that previously disclosed data. They were sufficiently interesting responses. We felt compelled to chase that signal into an expansion cohort, and so that's actually moved forward into that protocol. The second program, one that we have just announced that we've completed enrolling the first dose cohort, is the triplet program. That combines OX40 with 2 cytokines. A way to think of that is that we've actually added a little bit more gasoline to the overall immune reaction. It's mRNA 2752. And it's currently enrolling its second dose cohort, as you see there. And then just recently, we and AstraZeneca announced that we had opened the IND for the intratumoral IL-12 program. IL-12, a relatively classic immune cytokine. Anticipated next steps, obviously the OX40 program, we're excited to initiate the dosing in that phase 2 advanced ovarian carcinoma cohort, and we'll look forward to seeing that data when it's available. Triplet will continue our dose escalation and look for moving that eventually into disease-specific cohorts. Both of those programs will have arms that are in combination with checkpoint inhibitors. In this case, we're partnered with AstraZeneca, and so we'll be using theirs. And then IL-12, the phase 1 start, and that program is being run by AstraZeneca, as I said before. So that's the intratumoral immuno-oncology. I'll briefly touch on our localized regenerative therapeutics. This is a program that's in phase 2A, currently enrolling patients in CABBAGE. This is the VEGF program. And the goal here is to lead to cardiac regeneration in the context of cardiac ischemia. And so AstraZeneca has presented and published, and what I'm showing you here is a recent publication of the phase 1 data, which was done in patients with diabetes, was able to demonstrate both the safety, tolerability, protein expression, and bioactivity of the VEGF-A program. AstraZeneca has advanced that into a phase 2A. They're doing direct cardiac injection into the myocardium in patients undergoing CABG. And we'll look forward to the progress in that program and report it at the appropriate time once AstraZeneca has reviewed the data. Final two modalities very quickly, systemic secreted therapeutics and then the systemic intracellular therapeutics. And Lawrence described these. These are the far right-hand side of this chart. They will be chronic in many cases, although not all. And our first program that we took forward was actually a chikungunya antibody program. This is a relatively interesting technological hurdle. We're encoding an IgG, so this is a traditional monoclonal antibody. We identified this monoclonal antibody from a patient who had recovered from the chikungunya virus, and actually it's neutralizing to the chikungunya virus. And this is therefore a passive immunization approach. We're giving our drug, it's causing that patient to make the antibody, or the subject to make the antibody, and as a result of making that antibody, the subject would be protected against chikungunya virus infection in the future. We recently announced the study design you can see here, but we recently announced that we completed the first dose level cohort in healthy volunteers in that phase 1 study. Obviously, the IND is open and running, and we're looking forward to continuing to escalate those doses, and we'll be providing updates in the near future. With that, we then are excited to move into the other systemic secreted therapeutic programs. I will skip over the preclinical data in the interest of time for the chikungunya program, but obviously it demonstrated what we thought was really beautiful linear dose-dependent pharmacology. The programs that we're anticipating moving forward with relatively quickly here, so chikungunya antibody, we're looking forward to the phase 1 safety and serum antibody levels, but the two programs right behind that are a program for Fabry disease, where the next step will be an IND filing, and a program in another form of heart failure. This is a relaxin program that we would be jointly developing with AstraZeneca, and the next step there will be looking forward to IND filing. Last modality, very quickly, is the systemic intracellular therapeutic modality. This is very, very related to the last one I just talked about. And so whereas chikungunya, Fabry, and relaxin are secreted proteins, these would be making proteins for intracellular use. It is, once you're making the protein inside of the cell and causing it to be secreted, it's simply a matter of changing its localization sequence to direct it to a different part of the cell. In this case, our most advanced program is the methylmalonic acidemia program, 3704, where we had opened earlier this year the IND. We're given to a safe to proceed by the FDA. We subsequently were given a fast track designation for this indication by the FDA, and we're in the process of starting up those clinical activities and excited to move that forward. I'll remind you that MMA is a devastating disease with quite substantial morbidity and mortality through the second decade of life, for which there are no other therapies in development at this time. MMA and PA are highly related. So the PA program is propionic acidemia. It's another organic acidemia. And we have a natural history study ongoing there where we've recruited 32 patients to date. And so obviously we've been able to access these patients, about 20 MMA and 12 PA. I won't cover the preclinical data, but we think it's incredibly compelling for what's the potential of this medicine. Next steps are safety and proof of concept for the phase 1/2 data out of MMA. We'll look forward to filing the propionic acidemia IND, as well as a third program in this space that we have, which is the PKU program, which is earlier in preclinical development. With that, I'm going to actually turn it over to Lawrence to close.
Thanks, Stephen. I'll just wrap up with a couple of quick slides. These are all also going to be available on our website if you care to take a look. As far as financial results, I'll just touch on our cash. We had $1.7 billion at the end of last year. We recently guided to finishing this year with $1.15 to $1.2 billion. I'll skip over our priorities. This is a summary of all the next steps that Stephen just walked you through, which are all compiled onto one page. We view the next 12 to 24 months as an important time for Moderna as we generate clinical data across just about every one of our modalities and just about every one of our programs. Two things that I will highlight. We expect to do a science day coming up on May 7th in Cambridge. We'll be reviewing a good chunk of our research, which we don't usually discuss, but we'll provide an update on what's happening in the laboratories. And then in September in New York, we'll host an annual R&D day where we'll go through a comprehensive review of where we stand on our pipeline. More details to come on those, and then this is just a summary slide. I won't take you through it. You can have a look, but we are excited about the pipeline where we are, our presence across multiple therapeutic areas, and our ongoing execution and the scale with which we are operating and going to aim to continue to deliver. So thank you so much.